Hello, everyone. Good morning. I'm Professor Nada Bustani, and it is my pleasure to introduce the keynote speaker of our event today, Ms. Vita Lanoce, CEO of Lanoce Americas. Vita Lanoce is one of our very accomplished alumni. She earned her master's degree in biomedical engineering in 1985 under the guidance of Professor John Lee. Vita Lanoce is an accomplished executive leader in the life sciences and clinical research industry, with more than 30 years of achievement leading international clinical drug development programs and providing senior leadership for startups and mature organizations. As one of our highly accomplished alumni, Ms. Lanoce won this year's Rutgers School of Engineering Medal of Excellence Award in Research. This honor recognizes her distinguished contributions to clinical research and innovation and will be presented to her in the fall by Dean Tom Ferris. Ms. Lanoce is dedicated to realizing the vision and mission of improving health and well-being through innovative and breakthrough treatments in multiple areas of medicine. These include oncology, central nervous system pain, vaccine development, and general medicine. She is currently the Chief Executive Officer of Lanoce Americas, where her primary responsibilities are growth and oversight of the China and America affiliates of Lanoce Group, a Japanese headquartered global clinical research organization. After graduating from Rutgers, Ms. Lanoce began her clinical research career in academia at the University of Pennsylvania in the cardiovascular research team. There, she was one of the contributors who defined the heart failure model that is used for preclinical drug testing, with a focus on digitization of biological data. Ms. Lanoce then brought this model to Bristol Myers Squibb, thereby providing a robust preclinical pathway for drug development in the cardiovascular area. Within the clinical division of Bristol Myers Squibb, at the time as senior clinical scientist overseeing the cardiovascular division for drug development, led to the FDA approval of Monopril, a very well-known drug for the treatment of hypertension and heart failure. Her work led to refereed publications, and her research was selected for presentation at the American College of Cardiology. Ms. Lanoce is actively involved in industry organizations and initiatives, and she's an ardent advocate of health and fitness. The Helping Early Research Oncology Initiative, HERO, was formed under her leadership to bring awareness to the importance of patient participation in oncology clinical research. She is a member of the American Cancer Society's CEOs Against Cancer. She also supports a culture of wellness and fitness in her companies and in her community, sponsoring and participating in various wellness events, including, for example, the Horrible 100 cycling ride in Florida. She's a member of BioFlorida, bringing awareness to the importance of clinical drug development and the need for legislative support to ensure that the life sciences industry continues to be a leader in innovation and has the support for this. On the non-clinical research side of life, Ms. Lanoce is a private pilot and an accomplished flutist, receiving state and national recognition and the honor of participating in several youth symphonies, including Tanglewood, Rome Festival Orchestra, and Broward Youth Symphony. So we're really absolutely honored and thrilled to have her with us today. As the CEO of Lanoce Americas, an award-winning clinical research organization for excellence in the vaccine development industry, she's really poised to share with us today a unique and insider's view of vaccine development in the midst of a pandemic. Her talk today is entitled Pivoting in a Crisis: The Race to Develop Vaccines, the Approach, the Players, the Economics, and the Future. But just before we begin, I just want to alert everybody that we will be muting all your microphones that we have done already to avoid audio feedback during the presentation. There will be a way for you to participate in the presentation, and that's by putting your question in the Q&A box. So on your windows, if you see all the buttons there, the ... button should have a Q&A button inside of it, and that's your way to be able to ask questions. We will be fielding questions at the end of the talk. There'll be time for a Q&A session, but feel free to ask your question at any time during the talk. The questions will be saved in that box. So with that, take it away, Vita Lanoce.
Thank you. Thank you so much, Nada, for the wonderful information that you provided. What I'd like to do now while I am sharing my screen, hopefully everyone can see the presentation, Pivoting in a Crisis, the Race to Develop Vaccines, the Approach, the Players, the Economics, and the Future. I wanted to start out by giving you sort of a landscape. This figure is a very interesting figure where in the 1980s, the world map was drawn with the predominance of newly emerging infectious diseases. What we see today, this is what we have today, and this is from 2017. Currently, the SARS coronavirus is not on this list, but this coronavirus that causes COVID-19 was genetically sequenced and published on July 11th of 2020. This was the trigger point for our global R&D activity to develop a vaccine against the disease. The economic and scale of the humanitarian impact of COVID-19 pandemic has led to the focus on next generation vaccine technology platforms. These novel platforms pave the way to accelerate vaccine development. The first COVID-19 vaccine candidate was tested in healthy volunteers beginning March 16th of 2020. This is an amazing rapid response for our industry. So the novel SARS virus, we currently know is the virus that attaches to human cells by gaining entry through the spike S protein and replicates. It mutates fast, and there are approximately 40 strains at the present time. More importantly, there are two types of variants. These two variants are either quite deadly or they can produce a more mild, moderate infection. There is one key gene difference between SARS-1, which we had seen earlier, and this new novel virus SARS-2. We are not currently certain why that was triggered, but it has created a difference in behavior from SARS and MERS, as well as Ebola. In the prior infections, these diseases had no asymptomatic phase, which allowed for the contact tracing to be very effective. SARS-2, however, exhibits a high viral load early. This unfortunately helps with the spread, in addition to the spread caused by the asymptomatic carriers. Carriers have no symptoms spreading the virus unknowingly, which is why this got out so quickly. The key now is to suppress further replication, and vaccines are being tested now to that end. Containment is a priority. Implementing, which we have done, wearing masks, cleaning, distancing, all of which are our best defense at the present time until a vaccine and other treatments are available. We don't fully know the disease well at this time. The viral genome was sequenced within weeks, but there are still many unknown components. I know this looks a little bit like an eye exam, and I do apologize for that, but we're going to walk through the approach of how vaccines are developed. So a vaccine exposes the body to an altered and more safe version of a virus. It prompts our immune system. Our immune system produces antibodies, and those proteins will stop the real pathogen or suppress the real pathogen from infecting cells.
Vita, can I interrupt for just 1 second?
Yes,
You have a reminder that's blocking the screen? A WebEx meeting reminder, there you go.
There you go, sorry about that. So there's a range of technology platforms being evaluated. DNA and RNA, the nucleic acid, virus-like particles, peptides, viral vectors, recombinant proteins, live attenuated viruses, inactive viruses. These are all different approaches to be used. Many of the platforms are not currently the basis, however, for commercial vaccines, but experience in fields like oncology is really encouraging these areas, and researchers have pivoted to utilizing these opportunities that the next generation approaches offer, primarily increasing the speed in development and manufacture. This is what is critical in our future, for our future. It is conceivable that vaccine platforms may be better suited to specific populations as well. Elderly, children, normal adult populations. Everyone reacts differently to vaccines. And this is why we do so much in the field of research. It's not one size fits all. Many of you are probably familiar with the traditional platform, or you may not be, but we incubate the viruses in an egg platform. We use eggs for that. That takes a long time, and that has typically been how the influenza vaccines have been produced. The new cell membrane platforms speed up manufacturing process, and it provides an alternative to utilizing, the older methodologies. So let's talk a little bit about what this process is about. We isolate a virus protocol. We map the viruses for their genetic sequencing. That genetically engineered virus is then captured as their blueprint, and we embed that into a DNA plasmid or an RNA lipid or as in an adenovirus. These are then put into a solution so we can begin testing. The more traditional methodology to adapt it for a vaccine would be to weaken the vaccine or attenuate the vaccine, or a killed virus, an absolutely inactivated virus. As another option is to use the isolated pieces of the virus, those subunits, which we had just talked about, that S protein. Those are put into a solution. Following that, we start preclinical trials. This is very important because we want to make sure in lab cultures, we can see that there is some reactivity so that we are not just testing things that don't have any reactivity with COVID-19. And then the next important process is to ensure it is not toxic. We answer these questions. If it is, we stop testing. If not, we move forward with ensuring that we answer the question, does it produce an immune response, and attach the virus? So if not, we stop the testing. If yes, then we can move to clinical trials. This process, while can be very, the activation of this can be very quick in many ways, what happens is we don't see the responses or we have toxicity. So we want to do this part to be sure whatever we're going to put first in human and in clinical trials is safe. Our next step then is clinical trials. For the phase 1 portion of a clinical trial, right now this is what we consider first in human, if this is the first time we are testing it. We're asking similar questions. Is the vaccine safe? Are there side effects? Can these be avoided? And do we get an immune response? Notice here that the number of people tested is typically pretty low. And with our COVID, we are starting at the low end of the spectrum. The reason being, we get answers quicker. If this is a no, we again stop testing. If it is a yes, we can move forward. Phase 2, we're asking some similar questions. Is it safe? Do we get an immune response? Is that immune response strong enough? Because we may have an immune response, but if it's just a blip, It's not really doing what we need it to do. It needs to be strong enough. And this is where dosage really comes into play. We will do a lot of work in this phase to be sure that we have an effective dose, but we don't push the effectiveness to make it unsafe. Here we are testing in hundreds of patients. And again, there'll be a no or a yes at this stage as well. Many drugs fail in these stages of clinical research. And then phase 3. This is where we test tens of thousands. We make sure across a large swath of people and worldwide, this will need to be done in order to ensure ultimately that this is a safe and effective vaccine. And then traditionally, if this is a yes, it must be approved by regulators, the FDA here in the United States, the EMA in Europe. There's similar regulatory agencies in Japan and in China and all around the world, every country. Once that happens, then in parallel, that is typically manufactured in volume and then mass produced for the product to be distributed. Well, what's the difference today? The difference today is we have a much more rapid process through here. We can combine some of these phases. And what we have done and what the regulators have looked at is taking the production phase and moving it into this phase. So now this is risk. This is where the risk comes in. We could still have a no on many fronts here. But if we have good positivity in these areas, they're taking the risk of moving production forward. And that will help us be able to produce vaccines more quickly to meet the very, very extreme need that we have at this point in time. So who are the players? I have a listing of players here, and this is just scratching the surface. These are the players that in fact have vaccines in or near the clinical development stage of human testing. So what I think is very remarkable about this, Moderna started clinical testing of its mRNA-based vaccine just 2 months after the sequencing, which is amazingly quick. Recombinant platforms, as I had mentioned before, they are faster to create. They don't take months. They're safer, the vaccines, than the live vaccines or the live attenuated vaccines. And because this is such a virulent vaccine, these other methodologies to keep safety is really very, very important. However, none of these are this platform is technically not in commercial production. Moderna, however, may be our first to become a reality. Vaccines are based on viral vectors. Those typically offer a high level of protein expression and long-term stability. They're also known to induce strong immune responses. And finally, they are already licensed vaccines based on recombinant proteins for other diseases. So these set candidates could take advantage of the existing large-scale production capacity that we have. And that is very important. Many small vaccine companies, small biotech companies, they do not have the wherewithal to be able to ramp up. And they will need to partner and have partnered with big vaccine companies to do that scale up. Even the universities, AstraZeneca and University of Oxford have partnered. We have, of course, the government players, Pfizer, Inovio. I mean, there's so many amazing collaborations and partnerships that have come out of this, which is so very important. We still have over 115 promising vaccine candidates in the development pipeline, which is huge. But remember one of the things that I said earlier, in order to accelerate, we don't have the luxury of prioritizing. We must do things in parallel and continue to move these candidates through as much as we possibly can. Another coalition that I wanted to mention is CEPA, Coalition for Epidemic Preparedness Innovations. CEPI works globally to join collaborators to support and fund promising research. And our company, Clinical Americas, has formed collaborations with academic institutions, funding sources, as well as our services to launch startup companies to test novel and repurposed compounds for vaccines as well as treatments. We're currently working with several companies, each having either a single vaccine candidate or treatment candidate. Some have multiple preclinical candidates, and they're very promising. And we have a timeline of getting them into first in human trials in the next 60 days. So it's something that's moved very, very quickly. So the pipeline of vaccine candidates, how does that play out? We talked a little bit about the technology platforms. The recombinant platforms, the peptide-based platforms, the DNA and RNA. These are things that are really moving forward very, very quickly. And I thought it was very interesting that to see that developers in the private industry, as well as academics and public, this is how this work plays out at the moment. Private industry obviously was able to pivot very, very quickly to move forward to collaborate. And in North America alone, we have quite a bit happening with partnerships and those collaborations. So why so many studies? We have over 2,000 clinical trials globally. There's just an amazing amount, and this is both vaccine and treatment candidates, because we truly do need both. Well, again, I talked a little bit about this shotgun approach. It takes a long time to develop vaccines. Even with the innovations to shorten timelines and accelerate development pathways, mutations of the virus occur along the way. So this is another reason speed is so essential in this crisis. Different approaches provide pathway to vaccines which provide best immune responses. So we need to try multiple pathways. Ebola is another disease that is lethal, but it killed so fast, the local outbreaks initially did not have time to spread beyond the villages where it started. While this contained the initial spread, cities were still affected as villagers moved out to escape the spread. Clinical was among the first responders during this outbreak, sending research staff to Sierra Leone to test at the epicenter of the crisis. We had many health professionals, volunteers that wanted to be part of this research, and ultimately deployed approximately 40 staff members over a 3-month period. The economics and the disruption. I added disruption because I believe strongly that what has happened has been truly remarkable. The number of disrupted clinical trials is fairly staggering. This has affected companies, and it's a metric that we all need to really understand. It's affected organizations in industry and academia, those companies that sponsor clinical trials, collaborators, CROs, such as ours, and so many drugs are involved. And the majority of this, as you can see, nearly 30% are oncology trial delays. Another big area is central nervous system. These are two very, very affected areas. As we have all been in a situation of stay at home and only essential visits and procedures in healthcare, so too have the clinical trials been developed. But as an industry, we have to start looking at things differently. How can we restart, be safe, because so many people are in need. This cannot be put on hold forever. This is where the industry as a whole has adopted major changes. We have gone to end-to-end electronic solutions. Telemedicine has been incorporated. Everything you can think of electronically, we have implemented in a very short period of time to restart what we do. The pandemic and our response to change will forever change how we do business. So the future, from disruption to innovation. Disruption of the fundamental change from the traditional vaccine development pathway. It typically takes 10 to 15 years to develop a vaccine, which is staggering when you think about that. It's an imperative for scale and speed, which is unprecedented. So how do we do innovation? Our parallel and adaptive development phases, our innovation in regulatory processes, and scaling manufacturing capacity through collaborations with small and large pharmaceutical companies. Vaccines could be available under emergency use or similar protocols by early 2021. I wanted to throw this slide up largely because it was interesting in the beginning when Dave, we had the conversations earlier today. There are so many things that biomedical engineers can be a part of that are so important. I've highlighted adjuvants. There is an entire industry and specific companies working around adjuvants to improve immunogenicity and reduce the effective dose of vaccines. Very, very important. The nanoparticles that we heard talked about a little bit earlier. These are, types of formulations that improve vaccine effectiveness and acceptability. And electroporation, all of the new platforms, DNA platforms, etc. These are very, very important aspects that we really want to ensure that we are we're innovating in these areas to be able to best utilize these new platforms. So I'd like to just congratulate everyone on the phone. This is just an amazing time for you. I recall this day when I graduated. I was very grateful for the opportunity to learn. I was ready to start my journey. I took a very unconventional path after starting bench research by going into the pharma industry. And I took opportunities as they came to further learn and evolve. What I believe was instrumental for me was my time at Rutgers BME, which I do love best. What was it? The best ever program. I learned the art of seeing complex problems and breaking them down to their components to find solutions. BMEs gain a multidisciplinary background. It's foundational to see rapidly through complex issues. So to my fellow BME colleagues on this call, be open, be present, and be involved to find solutions to the many issues we have facing our future. We need your creativity and your innovation. Your journey will be written by you. So take opportunities to grow and learn every step of the way. Thank you very much for your time today. So,
I wanted to give everybody a chance to clap. Everybody for a minute. Thank you, Dr. Lanoce. I'm going to pass it to Dr. Bustani to moderate Q&A, and I invite all the participants to submit any questions for Dr. Lanoce.
Yes, and I just want to add again that the way to do that is through the more options ... button at the bottom of your screen. So I can start here with, in fact, a few questions that have been that Dr. Schreiber has contributed, and we've been wanting to ask Vita these questions. So how long do we usually have to wait to make sure that a vaccine is safe? And is that time changed now? So I know we have to develop everything very quickly, but does that also shorten the lifetime on our assessment for safety?
Safety happens along the way. So we start in preclinical with safety and we go through all of the phases with safety. In phase 1, that is where we really do begin. And even though we've shortened the timelines, safety is of paramount importance. So we are consistently looking at, when you think about safety, you look at, well, how many adverse experience are people having? Is there anything that we're seeing as a signal? And, statistical analysis helps us as well. So anytime there are any types of safety events, those will be rigorously looked at, and we stop the research very quickly when that happens. This is another reason why we have so many candidates through, because there will be an end to studying a particular candidate fairly quickly if we're seeing any toxicities or any safety issues. As an industry, we really work to ensuring that a well-controlled clinical trial gets to an ability to ensure, as best as we possibly can through this process, we look at the safety aspects. What we will do as well is vaccine research in this area will probably continue for many, many, many years. And those will be things that we will ensure that we continue to look at. So early research will help us, but we will need to continue that to ensure long-term safety, even as the virus mutates, etc. Influenza vaccine research has been an ongoing research for tens of years, because it is very, very important to continue that research.
Thank you very much. There is another question here from, I think, Kedar Patel. And the question is, do you think that moving up production would have some sort of pressure on lowering the criteria for a yes to move forward with the different phases?
That's a very good question. And I think that that's where everybody is sitting. How do we weigh this? What is the importance of getting a vaccine as soon as possible? I think that everyone is moving into this with the understanding that there is a huge amount of risk to start the production. But I believe strongly that through well-controlled trials, really focusing on safety, there will need to be that type of strong decision making process that we also have a very open and visibility into what's happening with these drugs, and FDA as well as the government support that we have through NIAID, through NIH, BARDA, and all of these organizations. We have an obligation to be sure that if we need to stop the production, it has to happen. So I didn't get into the stages of manufacturing, but the stages of manufacturing, there will be cut points that will be put in place in order to ensure that we can make those appropriate decisions at the appropriate cut point.
Thanks again. Another question here. How much money do companies stand to lose by manufacturing early in the timeline?
It's another good question. And I think that there's going to be I won't probably be able to give you an adequate answer on an actual financial number. What I can tell you is typical vaccine, and this is not just manufacturer, but it can be anywhere from 200 million to billions of dollars to go through the entire clinical development process, through to manufacturing. It's a very big undertaking. What I can say is that with large companies behind this, with the governments supporting, and it needs to be a global, worldwide investment versus it being a company, one company here, one company there. This is a global situation. There will be losses from this, certainly, because not every vaccine will work, but we believe strongly that the best candidates will emerge, and we will have those options for our future.
Thank you. I actually had a question about, testing vaccine, because once you get vaccinated, you need to be exposed to the pathogen to be able to test the efficacy of a vaccine. And I wasn't sure what the ethical responsibilities are of exposing people to the virus. And if that's not possible ethically, does that mean that we need the disease to remain prevalent in order to, in fact, be able to develop the vaccine? And that timeline is also important.
That's a very good question, because I really did not go into the mechanics behind the clinical study that's actually done. What we do is we will be checking, we will be taking blood from the volunteer. And it's the blood that will actually be tested for the titers that occur in, how we're making the antibodies. And then we will take whole cells. We will do a testing on those cells to see how they react to the COVID disease. So that's how the exposure actually happens, to determine the immunogenicity of the drug, as well as the effect, how effective this drug is. So it's not an actual exposure of the person, but it is of we draw the blood, and that's what we expose to determine whether or not that vaccine is effective.
Okay, and that's also for phase 3?
Yes.
Oh, okay, thank you. David's asking, is this really a race, or will there be only one winner?
I don't see there being one winner. I think that we likely will have and need multiple vaccines. And largely it happens that vaccines are not a one-size-fits-all. Our vaccines typically are, in adults, vaccines work a certain way, but you've got your elderly population where the vaccine could work differently, or it could be a different effectiveness, and you have to modify. And that's the same thing for infants and children and adolescents. So we also have to think about compromised people, comorbidities, people that have underlying disease conditions. As we've seen with this particular virus, those are the people that are at the highest risk. So what we're trying to do is to look at all potential options. Remember also, the virus mutates. It's how it lives on, how it spreads, and it becomes used to an environment, so it has to change so that it can continue to replicate. So we're always going to have some ability to look at a variety of different ways that we can attack this situation.
Thank you, Vita. So there's a bunch of questions here from students attending. I'll take them. Okay, here's a question from Sunshine Little Creek. Will use of plasmid DNA vectors for vaccine development, with use of plasmid DNA vectors for vaccine development, is anyone using multiple variant gene sequences for SARS-CoV-2?
That's an excellent technical question. What I would like to do is, I have invited our infectious disease expert, Dr. Joan Drucker, to this call. I think she may be better suited to answer that question. Joan?
Great, thank you, Vita. I'm an infectious disease physician who's been working in other emerging diseases for, for most of my career, which goes back quite a way. This is an RNA vaccine, so we would not be using DNA vectors for that, but this principle would be the same. Right now, I'm not aware of any of the over 100 things in development that you've seen listed using multiple gene sequences. They're focusing on a couple gene sequences specifically around the binding site for SARS-CoV-2. That seems to be the one that's the most antigenic for now, the most likely to succeed. There are certainly other targets. SARS is fairly simple virus compared to some others, so there are a few target genes that might work. Beyond that, so I'd say, I'm pretty sure the answer is no. It's hard to insert multiple genes at a time. That's possibly something someone would go after in the future, but unlikely unless the first targets for the spike protein fail.
Thank you, John. Here's a question on the FDA process from Stephen Segal. After vaccine has been developed and works effectively for COVID, do you think the FDA should tighten regulations or remain as open as it is now? Or remain as open as it is now, given that it's an emergency?
Sorry. FDA hasn't changed its underlying regulations. It is things for priority to speed review and do things of that nature, but it's not shortchanging any of the underlying either human safety conditions or manufacturing conditions, but it's a priority of review. So there isn't, and also has standards for vaccine developers to do. So there are places you can speed up the process. Some of the places you can't speed up, for instance, and why the shortest time frame that you may have heard.
And related to this, a question from Jay Sai is the FDA allowing more risk, and are the changes that the FDA has had to do to adapt to the pandemic, are they for the better? And do you foresee that there might be systemic changes with regulatory bodies as a result of this?
I think I'll take that one. I think that what we're seeing is an ability to be able to adapt to a crisis situation without taking away the safeguards that we have in place, because there are areas that we are looking to adapt and we are changing. I think that with anything in terms of a crisis happening, we identify those areas that we can make a change, and FDA will typically put out draft guidance, and this is what I believe will happen. Draft guidance will go out for public opinion, because we will learn a lot from this situation and how to adapt, what should be maintained, and where we can modify for the future, even in an emergent or situation where it is a crisis like this. So it is incumbent upon us as as researchers, the FDA, and regulators to really look at this for the future to see how things can change. But again, I think that something that Joan said that is very, very important that I'd like to be sure we get across is those safeguards that were put in place are still there, are still there. It is review timelines for getting things into the clinic, from preclinical, et cetera. It's just been a reprioritization which if you think back of what I was talking about before, that's why so many other clinical trials have been disrupted, because the entire focus has been on COVID-19.
Thank you. I'd like to take one, perhaps one last question. I'll see with David with the time, but this is an interesting question from the point of view of the public as well, is do these vaccines have a shelf life? If we start production before clinical trials are complete, do we run the risk of vaccines expiring before the clinical tests are complete?
That's an excellent question. You know, typically there is stability testing, and it's part of the manufacturing process. That stability testing goes to what your question is. But what we can do is many vaccines are temperature controlled. You can extend the life of this by keeping them in you know, a frozen state, etc. So when lots of the vaccine are produced, they always come with an expiration date. That will all be part of the manufacturing process that we are going to be discovering. So it's, I think that there's, if conceptually you can think about, it's not going to be manufactured in its totality in, you know, in a month. It's still going to take that entire, you know, that time frame. And there are components of how this is going to be manufactured so that at any given point in time, we will know what the stability is, how long it will be active, and how to best handle the vaccine in the future. So that's I think that it's a great question that is going to be managed as they manage the manufacturing process to ensure that we have what we need at the time that we need it, with enough of a shelf life to be able to be deployed as needed.
Thank you very much. I think what I'd like to do right now is I'm going to read one question that Joan can answer on the Q&A so that we can keep up with our time frame here for the event. It's a question from Brian Ayers. I just want to kind of acknowledge the students' participation here also in this presentation. It's great to hear all these voices. So zootransmission of diseases may be increasing from our impact on climate change. Do you see this pandemic changing or amending the clinical trial process? For example, more parallelization to accelerate addressing future novel diseases? And moreover, do you see a need for that? So, Joan, if you wouldn't mind answering this question by typing it offline, by typing your answer offline, everybody can actually read the answers to these questions as well. And so with that, I think I want to ask Vita for being with us this morning and sharing with us these very interesting thoughts about what's going on behind the scenes for vaccine development. And I think, I don't know if you want to unmute us, David, but I'm going to clap over here. We'll all have a lot of audio feedback. And also say that we have Vita Lanoce's contact information, and perhaps we can, I don't know, we could copy it back into the chat button or something if people have further questions, but your contact is on our flyer, I believe, or in our, BME database. So thanks again.
Yeah, it was my pleasure. Appreciate the invitation, and it was a pleasure. Good luck to every single one of you. You've got a lot that we are going to be looking to helping, for you to help us with innovation. So thank you so much.